This is a video explaining how the renin-angiotensin-aldosterone system works. That is quite a mouthful, so we'll call it the RAAS system. The RAAS system is part of the body's mechanism for controlling mean blood pressure. As the name implies, the main components of the system are renin, angiotensin, and aldosterone. This is how it works. Let's start with a box representing the desired blood pressure. When the blood pressure falls below that value, it is detected by the juxtaglomerular cells of the kidney. These cells are near the afferent arterioles of the nephron, near the glomerulus. A protein called angiotensinogen is constantly being made in the liver and released into the blood. When the blood pressure falls, the juxtaglomerular cells cause the cleavage of prorenin into renin. Renin is released into the blood. It acts as a proteinase, breaking up circulating angiotensinogen into angiotensin 1. Angiotensin 1 circulates in the blood and is cleaved by angiotensin-converting enzyme, ACE, into angiotensin 2. ACE is concentrated in the vascular endothelium in the lungs and the kidneys. Angiotensin 2 acts on receptors, angiotensin receptors. There are two types of angiotensin receptors, angiotensin 2 type 1, or AT1 receptors, and angiotensin 2 type 2, or AT2 receptors. The main kind of receptor in blood pressure regulation are the AT1 receptors, although there is also evidence that AT2 receptors also play special roles. Through AT1 receptors, angiotensin 2 does three things. It causes vasoconstriction, it causes the reabsorption of sodium, and it causes the release of aldosterone from the adrenal glands. It causes vasoconstriction by activating AT1 receptors on the vascular smooth muscle. The AT1 receptors are G-protein coupled receptors coupled to GQ and therefore cause IP3 production, which acts on IP3 receptors to bring about the release of calcium into the interior of the cell. This stimulates contraction in the vascular smooth muscle, which results in vasoconstriction. This vasoconstriction increases the mean blood pressure and the total peripheral resistance. AT1 receptors in the proximal convoluted tubule of the kidney cause increased activity of the sodium proton exchanger. This increases sodium reabsorption, and because water follows salt, it increases blood volume and hence mean blood pressure. As we have already seen, angiotensin 2 stimulates the release of aldosterone from the adrenal gland. Aldosterone acts on aldosterone receptors in the collecting duct of the nephron. This causes sodium channels to be inserted into the wall of the collecting duct, and it also causes more sodium-potassium pumps to be inserted as well. Both of these things work together to increase sodium reabsorption. So you can see that the renin-angiotensin-aldosterone system is distributed widely around the body and involves many organs: the liver, where angiotensinogen is produced, the lungs, where angiotensin-converting enzyme is concentrated, the vasculature, and the adrenal glands, to name a few. The work of the renin-angiotensin-aldosterone system complements that of the sympathetic nervous system. Whereas the autonomic nervous system responds rapidly to moment-to-moment changes in blood pressure, the renin-angiotensin-aldosterone system is slower and makes longer-term adjustments. There are several sites in this pathway where drugs are applied to reduce the increase in blood pressure brought about by the renin-angiotensin-aldosterone system. Starting with renin, there are drugs that block the action of renin. An example is aldosterone. Aldosterone blocks the production of angiotensin 1 from angiotensinogen. The next step in the pathway the conversion of angiotensin 1 to angiotensin 2 by angiotensin converting enzyme, is blocked by, for example, captopril. Captopril resembles the last two peptides of angiotensin 1 and is thus mistaken, as it were, by the ACE. Captopril was one of the first drugs to be designed based on molecular understanding of the natural ligand. Next we have angiotensin receptor blockers, which bind to the angiotensin receptors, preventing their activation by angiotensin. The best-known class of drugs that do this are the sartans, such as losartan. Aldosterone antagonists, such as spironolactone, and sodium channel blockers, such as amiloride and triamterene, both serve to block the dual effects of aldosterone on the collecting duct of the kidney. These are therefore classed as diuretics, but they differ from other diuretics, such as furosemide and the thiazides, in that they do not involve the loss of potassium.